Support Microbe TV. It's time for our annual fundraising drive. We depend on your support to produce high-quality science videos and podcasts, and now is the time to help us. If our programs have appealed to your science interests, or if they have helped you in some way, please record a brief audio or video with your phone and tell us about it. Let us know how we empower your inner scientist. We'll use it for our fundraising efforts. Send it to incubator@microbe.tv. And for more information on how you can give us your support, go to microbe.tv/contribute. This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV. This Week in Virology, episode 1173, recorded on November 29th, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Madison, New Jersey, Brianne Barker.
Hi, great to be here. It is 43 Fahrenheit, 6 Celsius. Not currently raining, but I think it will. And nice day to be cozy inside, thinking about science.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 30 Fahrenheit, but it says it feels like 12. It is quite windy, and that's -3 Celsius. And we've had a little bit of snow, and it's kind of lightly snowing now, too. But it's not sticking to the streets or sidewalks, so it's just pretty.
From Austin, Texas, Rich Condit.
Hey there. 58 degrees Fahrenheit. I'm interested to hear from our other Texas guest. But that actually, as I discover opening my my weather app, that's 1 degree warmer than where I used to live in Gainesville, Florida. And this is going to be our high temperature for the day. We've finally exited summer for real, I think. And I want to apologize to those watching on YouTube for the fact that my image is washed out, but I can't do anything about it because Tinkerbell here is on my shoulder, and she's got to have her way. She's got to steal the limelight.
And from College Station, Texas, Jolene Ramsey.
Howdy. It's good to be here today. It's 59 degrees Fahrenheit here in College Station, and that's 15 Celsius, and windy. It's windy out there.
I can see it through your window. It's windy.
Mm-hmm. Yeah, look at that.
Oh, yeah. The squirrels have been pretty active lately, so,
I was going to ask
keep an
about
eye.
the squirrels. All right.
Check out our video to find out why RFK Jr. is not suitable for the Director of Health and Human Services. We're going to talk more about some other appointments today as well. And write your senators to oppose the nomination of any of these people, RFK Jr. and all the others. safecommunitiescoalition.org/RFK. It's a website put together by a 501c3 just for this purpose. That's just for RFK. I think they have to make separate ones for others. But the person who made it was in touch with me, so I'll get in touch with her.
If you have a newly elected senator, it will be automatically going to the previous senator. So I had to do an old-fashioned print out the letter and put it in an envelope to mail to the senator-elect from Michigan. Yeah, but I did that too.
We're now in the fundraising part of the year, November, December. Channel your inner scientist. Support Microbe TV. Go to microbe.tv/contribute. We have the usual ways that you can contribute, but there's some incentives if you'd like to do that as well. And the Rosenfeld Lab, says release the viral immunologist in you. Rosenfeld Lab in the Division of Viral Projects at CBER, FDA, is looking for a postdoc staff scientist to continue the lab's research understanding the immune responses elicited during enterovirus infection. If you're an immunologist, microbiologist, or virologist wanting to learn more viral immunology, email Amy at amy.rosenfeld@fda.hhs.gov for more information. What's up with ASV, Kathy?
Well, as we've mentioned, it's going to be in Montreal this summer, July 14th to 17th. And the website is asv.org/ASV2025. In fact, I think if you just search for ASV 2025, you'll get the website. But right now would be a really good time to renew your membership. Or if you're planning on submitting a travel award application and you're not yet a member, go ahead and get your membership taken care of now, and then you'll have that out of the way during that frenzy when you're trying to get your abstract submitted. So you can do that by going to the meeting website, click on Grants and Awards, and then drop down to Travel Awards. And then on the fifth line, there's a link that talks about membership here. So, or you can just go to the main website and search around for a membership. So it's also a good idea to be a member if you're thinking that you might use one of the childcare options, either ASV Cares or the on-site childcare. Both of those require you to be a member. So
I have a lifetime membership. I never have to do this again.
that's
So do
right.
I.
Good for you.
It's
So,
just-
Kathy, remind me, are you still it?
No. I'm just the PSA announcer now.
Okay.
Kathy's
Congratulations.
job was so big,
Thanks.
she got split into two people, right?
Yeah,
Two people
right.
are taking care of those responsibilities.
Congratulations, not only that, but thank you for your service. That's
Oh,
that is a huge job that you did. How long did you do that?
I did it for 7 years.
Wow.
Yeah.
I mean,
Yeah.
that's just a huge contribution.
Well,
Thank you, Kathy.
thanks. I had a really great team and I really enjoyed it. And part of the reason why we needed two people is that ASV has expanded its mission so much that it's just more than one person can oversee. And so we've also increased the ASV staff number from 1.5 to 2. So, yeah.
Good.
Yep.
Okay, a few news items, mostly having to do with more health and science related nominations.
Mostly
but
having to do with making me sad right before this episode.
this just bothers me so much, really. After 45 years in science, to see it, that people want to break it, basically. It's just, I just don't get it. These are, anyway, first one, the new NIH director. This is an important position. Jay Bhattacharya. Oh my gosh, there's an article in Science you can go read.
The science article is very good.
Yeah. He's at Stanford. He's a health economist, an outspoken critic of, the handling of the pandemic. And you know what? You can be critical. That's fine, as long as you do it based in science. But he didn't have anything constructive to say, in my opinion. But more importantly, he co-authored the Barrington Report and believes SARS-CoV-2 came from a lab. How can you be the NIH director and have done that? I mean, we talked about the Barrington Report at the beginning of the pandemic. It's horrible.
Yeah, if anyone doesn't remember the Barrington Report, it was the one that was like, let's let it rip and infect everyone right away.
Yep. Among other things.
As I read the science article, the thing that it just struck me as most disturbing was the support for the lab leak idea.
It's crazy. With no scientific basis or evidence, plenty of evidence for a natural origin, and he refuses that. And in fact, in this article, they say he emerged as a pundit on X, and that's the thing. The pundits were not telling the right stuff. He's advised Florida to drop masking in schools. Oh my gosh. Yeah. What else did they say here? Oh, my former student here. I have to read her quote. Angie Rasmussen always has pithy quotes.
Yeah, she came down pretty hard on this.
She wrote, "He's a self-interested extremist who gives cover to anti-vaxxers and will destroy NIH." Angela Rasmussen, a virologist at the University of Saskatchewan. Yeah.
She was a PhD student with you?
She was. I'm glad that I didn't evoke her ire in any way, at least not that I know of. She's not shy at speaking out.
What do they say?
And he said he was one of the people who said herd immunity would end the pandemic. You know, you can be wrong. That's it. But someone here says, Keith Yamamoto, who I have a lot of respect for, right? Biochemist at UCSF. He has contributed to erosion of trust in science by attacking experts.
That's one of the most disturbing things. Okay, you can disagree with the science, Okay, if
Yep.
you've got good analytically based arguments, but well,
Well, you got to be
extraordinary.
decent. You have to be considerate. You don't attack people, right? And that's what he did. Okay, so that's NIH director.
Well, the one thing that it says in here that Dr. Bhattacharya espouses is taking more risks with grant funding. So
What does that mean?
There were a few positive comments in this article.
But does that mean
was one of them.
Risking things like unproven non-antivirals like bleach?
That's an interesting spin on that. But one of the things I saw was that in encouraging funding of young and new investigators, okay, as a part of that sort of risk-taking thing. I'm all for that. That's
yeah,
fine with
that would
me.
be good.
I don't know who's going to get confirmed or not, but he has to work with RFK Jr., who's going to be the head of HHS, who wants to cut 600 NIH employees on January 20th, right away. And I'm sure he's not done any kind of study to see why. He just picked a number.
And posted some of their contact information on social media.
He did.
Really?
He picked 600 specific people.
Well, so he and Musk have both singled out people that they want to cut and have posted.
I don't believe that he's gone through 600 people. That's too much work. I don't believe it for a moment. The other thing is, of course, RFK Jr. wants to cut back infectious disease research for 8 years, which is the dumbest thing I've ever heard, honestly. Because today everybody's all upset about H5N1. You need to have some infectious disease research to know what's going on, right? You're not going to do it?
I think I heard that the reason he picked 8 years was this was when he was still running for president, and he was assuming that he would serve 2 terms.
That's right. He's still saying it, though. He wants to cut infectious disease research and focus on chronic diseases, okay? Because people are too obese in the U.S. They have diabetes, blah, this and that, and he's going to solve it. Good luck. In the meantime, we're going to have rip-roaring infections. And this is what gets me very sad, that people are talking about it. And half the U.S. doesn't seem to care. I don't get it.
Well, the trainees coming to talk to me walk into my office and say, well, I'm good, but I'm also sad because I read the news. And so it's definitely not doing anything to help us with the problems that we have in filling the needs and the bringing in new creative people. I think it will make them have less incentive to be scientists.
Sure. Mm-hmm. I mean, you have a wonderful science establishment in the U.S. and you want to break it. I don't get it. Just to be contrary. Okay, the next one. This one, I reach out to Amy, the FDA director, Martin Makary of Hopkins. I have a quote here. He wants to properly evaluate harmful chemicals poisoning our nation's food supply and drugs. But what are these chemicals poisoning our food supply and drugs? Does anyone know?
Chemicals. Chemicals are bad.
Okay.
Dihydrogen monoxide?
Yeah, that one is the worst. It's everywhere. Oh, my gosh. So this guy, another outspoken, Fox News. I can't sign in anymore because I've looked at it too many times. Fox News commentator. In my opinion, unqualified for this position. And CDC director, Dave Weldon. Let's see if I can get into this article. Dr. David Weldon, a former congressman. No, I can't get into that one. New York Times is
you have something just written in the doc.
Yeah, as member of Congress, Dr. Weldon pushed the false notion that thimerosal, a preservative that used to be in many vaccines and now are in just multi-dose flu vaccines, had caused an explosion of autism, for which there's no evidence. There have been dozens of trials, and there's no evidence for that. He also introduced a vaccine safety bill that aimed to relocate most vaccine safety research from the CDC, which he said had an inherent conflict of interest. How does the CDC they don't make any money. How can they have a conflict of interest? And furthermore, they make recommendations. They don't say you have to do this. And many state health departments depend on them. What is the CDC about vaccine schedules? And they take their advice. So I don't get this at all. This is being contrary for the sake of being contrary, right? This is the world we're in. In my opinion, every one of these nominees is a hack, including Dr. Oz, who we haven't talked about. We talked about last time. They look like they're okay, right? Stanford and Johns Hopkins, good places. But as soon as you look below the surface, they're hacks. They're not suitable.
I forget what Oz has been nominated for.
Medicare and Medicaid.
Okay.
And so you can get your snake oil purchased from your Medicare now.
There's no right to health care, and the uninsured can get checked once per year at a 15-minute thing where a bunch of doctors will go, and it will have a, I quote, festival-like atmosphere.
Great. Mm.
So, I don't know, people listening are probably already converted, but the others, this is what you wanted. And if you didn't know that this was going to happen, because there was a lot of rumblings beforehand, well, you need to investigate your candidates, right? But you didn't.
You can, no matter who you are, you can do a lot simply by speaking the truth to whoever you're in contact with. I'm actually in contact with quite a few people who who are either not necessarily well informed about this stuff, or might have opinions different than my own, and just without being confrontational about it, you can just tell them what you think.
Yep. Okay, we have two news things about real science. First is an article in JAMA Network, which I also can't get to, but I'll just tell you the title. Cervical cancer mortality among U.S. women younger than 25 years, 1992 to 2021. And this is a study. HPV vaccine was approved for routine use in U.S. women in June 2006. And from 2022 and 23, there was a 12% per year decline in cervical cancer incidence from women less than 25. So because that should lead to less mortality, then they said, is it being translated into less death? And I don't know the outcome because I can't read it. Does anyone have it?
The answer is actually that, yes, it did lead to
decreased
fewer
mortality.
deaths.
Okay, good.
For the for those who are not necessarily up to speed with this, okay, we're talking about a papillomavirus vaccine that's specific for certain strains of papillomavirus that cause cervical dysplasia, some fraction of which go on to cause cancer. And these viruses, basically cervical carcinoma, which has been a real killer in the U.S. and is still a killer globally. These viruses are effectively the sole cause of that disease. So we have a vaccine that is effective against a cancer. But one of the things is that the cancers, we vaccinate people who, the best option is to vaccinate people before they become sexually active, okay, because they're sexually transmitted viruses. And there's pushback against that from people who relate the sexual activity to vaccination, blah, blah, blah. But forget that. You vaccinate people before they're sexually active. But you're not going to find out whether that was effective for decades, okay? Because it'll take 10 or 20 years before you know whether that has impacted the incidence even of the cervical dysplasia, and another decade or so before you know whether it influences the incidence of cervical cancer, and even longer before you know whether the mortality rate is impacted. So I forget how long it's been since these have been around. Probably
18 years.
18 years? Okay. So we're just getting to the point where we can make some of those assessments. Okay. And that's one of the reasons this is important. It's clear that the vaccines are effective, but now we're seeing it in real public health terms.
One of the other things that this mentions is that There's been between 2022 and 2023, 4%, more than 4% decrease in the vaccination rate among adolescents for whom this is most important. And so this highlights the urgency to improve vaccination coverage. It went down during the pandemic, or at least it didn't go up. And then there's been this troubling decline recently.
Yeah, so just to emphasize that, some of the misinformation that goes out is the notion that either I don't need to vaccinate my children because they are not sexually active, but that's not the point. The point is you need to vaccinate them before they become sexually active, because no matter who you are, the probability that if you engage in any sexual activity at all, that you're going to encounter these viruses is extraordinarily high. Okay, it has nothing to do with your morals or anything else. Okay. And then there's this sort of, you get this thing about how if I vaccinate them, it's going to somehow license sexual activity in them. And I'm sorry, wake up. People are sexually active no matter what you do. Okay? So protect them, please.
I think the other thing that I often hear is that I don't want to vaccinate my son because he doesn't have a cervix.
Right.
However, that notion is forgets a lot of things about how these are transmitted, and also HPV can cause cancer in men, just a different kind of cancer.
Penile, anal, genital, and
Head and neck
oral
cancer.
cancer. Oral cancers. Yep.
Yeah.
By the way, this is one of the vaccines RFK Jr. says is of no benefit and causes more harm than good.
Not
Totally
true.
wrong. Ignores
Not
the
true.
facts. Not true.
Actually, I have pardon me, I got to go on about this a little more. But some listeners with a history may know that my sister-in-law died of a cervical carcinoma. Must be 32 years ago now, something like that. When I was just starting to teach at the University of Florida and acquired the responsibility for teaching the medical students about papillomaviruses. And at that time, there were even in the scientific community still skeptics about whether or not the papillomaviruses, and it was, reasonable skepticism about whether or not papillomaviruses were the major cause of cervical carcinoma. And over my 25-year history teaching at the University of Florida, I watched this progress, teaching these lectures from just barely being able to say that the viruses cause this, to identifying the viruses, to distinguishing the high-risk viruses from the low-risk viruses, to predicting that maybe that understanding would help us do diagnostics for how much risk there was that this particular virus that you had would lead to cervical carcinoma. The one thing dumb me didn't anticipate was that would translate into a vaccine. And sure enough, a vaccine comes up. So this is a remarkable example of how science contributes to public health over the long term. And hundreds, if not thousands, of people involved in a very thoughtful research.
All right, one last item, and this I can get in. It's from Texas Health and Human Services. The first locally acquired case of dengue virus infection in 2024 in Cameron County. Now, normally, most of the dengue in the U.S. is travel-related. You go somewhere where there is dengue, typically Puerto Rico and other places. You come back, you get sick. It's travel-related. But this person had no travel history, so it was locally acquired. That often happens in near the border of Texas and also in Florida, where you have the right mosquitoes for transmission. Where is this Cameron County?
Yeah, I looked it up because I didn't know exactly where. Yeah, Brownsville.
It's the
right.
Rio Grande Valley. Okay, it's the
It is,
southernmost part of Texas.
It's about as far south in Texas as you can get.
Yeah,
it's on the Gulf and very, very close. Well, borders with Mexico, and then also close to Big Bend National Park. It's a very large county.
Unfortunately, I'm going to miss yoga tomorrow morning. where my instructor is an epidemiologist in Texas, okay, because she and I could have a conversation about this, but I got other things I got to do.
Okay, on to the literature. We have a snippet about influenza H5N1. It is it's a paper that has been accepted to Nature, and we have an accelerated article preview. So they put out the article, not formatted. It's just the submitted paper, and it's been accepted. A human isolate of bovine H5N1 is transmissible and lethal in animal models. So I think they should have virus in that title in some way, right? Because this is preaching to the cognoscenti, but what if other people want to know? Okay, so put virus in. This comes from the lab of Yoshikawa Oka, and we have 1, 2, 3, 4, a lot. Chunyangu, Tadashi Mamura, Li Zhengguan, Ami Eisfeld, Ashim Biswas, and Maki Kiso are all co-first authors. And they are from the University of Wisconsin-Madison, the University of Tokyo, University of Shizuoka, and the National Center for Global Health and Medicine Research Institute. So Kawaoka has positions at Madison, Wisconsin, and University of Tokyo. He goes back and forth every couple of weeks. Has more flyers miles than any of us. So this is a very interesting article. As all of you know, in the last couple of years, avian H5N1 influenza viruses, which infect all kinds of wild birds, particularly water birds, have traveled globally. Their seasonality is gone, so now they're infecting all the time. And they've come into North America, where they have established an infection in dairy cows. And 214 herds in 14 U.S. states have been infected. We talked about this before. The cows get mammary gland infections. They're really okay. They don't get very sick. But then the virus ends up in the milk and you can't sell it, right? And we did a paper previously by this group on the characterization of an H5N1 virus from a cow, right? And they found that you could give it to mice. It would kill them. It replicated in respiratory and non-respiratory organs in those animals. It could infect ferrets, but it didn't transmit very well among ferrets. But now this paper is about an isolate from a human who got it from a cow, right? There have been 14 cases of human infections of humans from cows, from dairy cows, in 4 different states as of September.
And this person got it as an ocular infection. And I had just listened to the TWiV from Brisbane, where Steph Langle and Jenna Guthmiller talked about milking cows. And of course, the udder is right at eye level. So it makes some kind of sense for the ocular infections.
Actually, if you look at the CDC update that you linked,
yes.
Vincent, that I really like, it counts 32 human cases from cattle, 28 from California, and a smattering from elsewhere, and another, just for the record, 21 from poultry. So the majority of those that have been scored, 55 human cases, have come from cattle.
So this is a case in March. A dairy worker who was working with sick cows got conjunctivitis, but no fever, no respiratory symptoms. And they isolated a virus from a conjunctival swab, which they call HUTX37-H5N1. So the point of this paper is to ask, has it undergone any changes after replicating in this human that make it more of a pandemic concern, right? So they do an extensive virological characterization in cells in culture, in mice and in ferrets. All right, so first they do growth curve analysis. You infect cells, you measure virus production over time. You include a time zero always. Does everyone agree that you need a time zero?
Oh, absolutely,
Yes.
man.
You know how much pushback I get from people who don't have time zeros? And I say, you can't interpret anything from that. Oh, it's too expensive, too many animals, blah, blah, blah. Too expensive is never an excuse. You got to do the experiment properly. You need
And
a
this
time
was just
zero.
for growth curves, right?
Just growth curves for many things, right?
Yeah.
Any time courses that you do.
I'll tell you, to me, this is really nerdy, okay? To me, the coolest thing about the time zero, first of all, I had to learn how you do the absorption, and then you got to wash the monolayers like crazy. So you're just looking at the viruses that are absorbed, okay? But without the time zero and a fine time course beyond that, you can't see the eclipse. And the eclipse is so cool. That's where the titer in the culture actually decreases because the viruses have been absorbed and uncoat, so their infectivity goes away, and then it comes back up.
You do it in animals, too. You have to have a time zero. So if you're doing 10 animals per time point, you have to have 10 at time zero. People complain about that. It's a waste. It's not a waste. It makes your results valid.
Well, actually, we're getting ahead of ourselves, but I think about this when I think about these ferret transmission experiments, because they infect the ferrets and then they wait a day and expose them to another ferret. And I'm thinking the whole time about the time course of infection. They wait a day so that there's not a lot of loose virus around, but do you really know that?
No.
Okay, what actually happens in a fine time course to the virus titer in those animals?
All right, so the first set of experiments, they take primary cells, epithelial cells from human, either conjunctival cells, that's the outer surface of the eye, or the lower respiratory tract alveolar cells, and they infect them with 3 viruses. So the HUTX37, here, the human isolate, a virus that never saw a human. It was isolated from the milk of a cow, and this is going to be called NM93. The hardest thing is to remember all these numbers. I always forget.
Well, if you think about to me, it was just like the Texas one is the milk human and has human in the name. So if you just say the human Texas, and then the New Mexico one is from the cow, and then the last one is from 2004. It's like it's a way back control.
Yeah, that's from an infected human in Vietnam. So that's an avian origin, H5N1,
right,
as opposed to a cow origin. All right, and so they do infections at 33 degrees Celsius and 37 degrees Celsius. And the human TX virus grows really well at both temperatures. And this is important because there is an amino acid change in the viral RNA polymerase of influenza viruses that allow them to grow well at 33 degrees. And so this virus, HUTX, has a change in the polymerase. It's called PB2627K. But the Vietnamese isolate from birds, from a human infected by a bird, also has that change, and it's not enough. There must be other changes in the TX isolate to allow it to grow at 33 degrees Celsius, and they all grow in corneal epithelial cells.
And we should probably remind people why we care about 33.
Oh, 33 degrees?
Yeah.
Well, it's the temperature of the upper tract in the eye, right?
Yeah.
So the virus has to be able to grow at 33. Not all viruses can do that.
And that might also impact, how transmissible the virus is.
Exactly. Next they do some experiments in mice. So they want to determine the mouse lethal dose, 50%, the amount of virus that will kill 50% of mice. So you inoculate mice intranasally with different amounts of virus. This is amazing. The 50% mouse lethal dose of HUTX is less than 1 PFU. I've never worked with a virus that's that lethal.
Yeah.
Oh, but we have. And what that means, to me anyway, is that the virus is, that the mouse is a better assay for infectivity than your cells in culture where you're measuring PFU.
Yeah.
Good point.
So, could be, yeah.
Good point, because once again, for the uninitiated, what we call particle to infectivity ratio of a virus, that is how many particles you have and how many infectious units you can measure, is usually pretty high. It can be 10, can be 100, can be even more. But a lot of that is, how good is your assay system? So good point, Kathy.
So the TX is less than a PFU. The NM, right, the New Mexico virus, 31.6 PFU, and the Vietnam is 2.2 PFU. So this human isolate is more lethal in mice than the cow isolate. So where does it grow in mice? Where does it reproduce in mice? So they infect mice and they take various tissues, all kinds of tissues. And day 3, they can find virus in all mice, in all tissues. This is with 10 to the 3 PFU. The highest amounts of virus in the respiratory tract, nasal turbinates, trachea, lung, but they can also find it in spleen and in blood, etc.
Now,
There's a lot in blood.
I was surprised. I've never studied influenza, and I hadn't realized before that if you took, every body part that they measured here has presumably above the threshold of detection because they're showing points, except for the colon at day 5.
Yeah.
So
it's a feature of H5N1 viruses in particular because they have a protease cleavage site in the hemagglutinin, which can be cleaved by furins, which are present in all tissues, as opposed to all other influenza viruses where the proteases are only in the respiratory tract. So you don't see that pattern with other influenza viruses.
Is it because but why are they there in the first place? Is it because they're viremic? It's distributed by the blood?
They can get into, if you're replicating in any tissue, you can get into the blood. The question is whether that plays a role in the pathogenesis, right? So for human influenza, it doesn't. You get a little viremia, but it doesn't if the virus goes to another tissue, it doesn't reproduce because it doesn't have the protease. But for H5N1, yes, it plays a role in the pathogenesis, and you get systemic infection, multi-organ infection. That's why it can be quite lethal, right? And also, they don't do it in this paper, but the New Mexico isolate also spreads systemically. So there's no difference from the human and the cow isolate, right? And then finally, ferrets. So mice lie, monkeys exaggerate, ferrets are not humans. So they previously, in the paper, we did this paper, NM virus from cows has limited lethality in ferrets and doesn't spread very well. So they take the Texas isolate, intranasally infect ferrets with 1 million PFU. All the animals died. They have weight loss, fever, and they all died. So it's quite lethal, which is unusual. They say later it's unusual for an H5N1 virus in ferrets, this uniform lethality. They find virus in all tissues. And you don't see that with NM, right? There's limited lethality in ferrets with the cow isolate. So somehow having gone through a human, now this virus is more lethal in ferrets. But remember, we'll get to this in the discussion, the person who had this infection was not very sick, had conjunctivitis, right? So that's why we say ferrets are not humans, right? Anyway, transmissibility in ferrets, NM didn't transmit well. So in this experiment, you infect a ferret, you keep it in a cage, and then you put uninfected ferrets in the cage next to it, right? And there's air passing through, and you see if the uninfected ferrets can get infected. And the NM virus does not transmit very well. But the Texas virus does. If you inoculate them with 10 to the 6 PFU, about a third of them can transmit infection. And if you use different amounts of virus, you can get anywhere from 17 to 30% transmission with 10 to the 3, 10 to the 2, or 10 to the 1 PFU. So it transmits better than the other virus.
This is a huge ferret experiment. Okay,
it is.
When they introduced the first experiment and did 1 million PFU, I'm thinking since it's only infectious at it's infectious down to 10 to the 1 in mice. I'm thinking to myself, man, wouldn't you do some sort of a dose titration first? And then I think, well, ferrets are expensive, so And 10 to the 6 will do it, so you do that. And then they finally, when they get down to this experiment, do the dose experiment, and they got 6 pairs in each of 4 groups.
Yeah.
Right? So that's like almost 50 ferrets. Holy cow, that's huge.
Yeah. Oh, this is infectious disease research. Did you know?
Oh, sorry.
Can't do this next year.
Right.
Gotta hurry up and do it.
What biosafety level do they have to do this at? Do
3.
you know?
They're doing all this at 3.
Okay.
I looked at the methods because
Thanks.
we'll get to this. They've also got a big rap about all of the hoops they jumped through to get approval to do this research. Okay, we can talk about that.
So far, the Texas isolate is highly lethal in ferrets. It transmits by respiratory droplets, and it seems to transmit better than the cow isolate. Although they didn't actually do the cow isolate in this paper, as that would be even more ferrets. Right? So they already had done. You could argue that you need to do it on the same day, right, to make conclusions. I don't know. They sequenced these viruses to see if there were any amino acid changes that could give them some clue about what's controlling these changes, right? There is a change in the polymerase E627K, right, which is this change always occurs when you adapt it into mammals from birds. It allows the polymerase to interact with a host protein, which Andy Mealy talked about in the TWiV in Wisconsin years ago, which I can remember the title, Science Will Break Your Heart, right?
It's only like 2 years ago.
It's only 2 years ago?
Yeah, that was a long time.
So this change is found in the human isolate, but not in the cow isolates, which is interesting because cows are mammals, right? But who knows? The cows have a different change, which they then show actually can confer higher replicative ability in human cells. So you have to always look for more than what you think is important. And finally, there are 2 more experiments. One is the receptor binding. The New Mexico isolate preferentially binds to alpha-2,3 linked sialic acid. So avian influenza viruses prefer to bind to alpha-2,3 linked sialic acids. And those are, in birds, influenza virus is a gastrointestinal infection, and the GI tract is full of alpha-2,3 linked sialic acids. It will also bind to alpha-2,6. And in humans, our upper tract has mainly alpha-2,6, except the eye, which has alpha-2,3. So that could be why you get often conjunctivitis with these avian oustes. And then if you go deep into the lung, you get alpha-2,3 sialic acids. And they say here, if a H5N1 virus acquires the ability to bind alpha-2,6, that's a risk factor in pandemic assessment, right? So this Texas HUTX isolate preferentially binds to alpha-2,3, but it will also bind to alpha-2,6 sialic acids. So that's a bit of a concern, right? So that puts it high on the pandemic risk assessment. And finally, they ask if the antivirals we have can inhibit these viruses. So we have neuraminidase inhibitors, and we have polymerase inhibitors. And one of the polymerase inhibitors is the endonuclease, which is part of the polymerase. And the unmodified virus, the TX isolate, they're all sensitive to these inhibitors. But there is a amino acid change that they found in publicly available neuraminidase sequences derived from a cow H5N1 virus, which confers some resistance to the neuraminidase inhibitors. And they show that if they incorporate that into the isolate, it doesn't protect against lethality in mice. So that's a bit of a concern. But the others, the polymerase inhibitors, those are favipiravir and baloxavir, are highly effective against Texas isolates. So that's good, right? I hope we have enough of them if this becomes a problem, because we're not going to be able to make more, right? Can't do any more research.
So I'm reading through this paper, and I see that this Texas isolate has this is nastier in ferrets.
Yes.
Causes more of a problem and is transmissible, and that the suspect mutation is this polymerase mutation. And I'm thinking to myself, okay, so the obvious experiment is to put that mutation in a virus that is not as lethal in ferrets and see if we can determine that mutation is what's causing that. And of course, my immediate thought is, oops, that's a gain of function experiment, okay? And that's probably a no-no. And sure enough, it turns out that when they get to testing that mutation, they do this reporter assay, a replicon assay, so they don't make virus. They look to see if that mutation actually confers higher polymerase activity in human cells, which doesn't really address the fundamental pathogenesis question. And then I look in the methods, and this is why, Kathy, I knew that they were doing this stuff at BSL-3, because the methods have a big rap about how everything went through all of the appropriate approval channels, including the local dual use research of concern panel and et cetera. So, you know damn well that they would have considered doing that experiment, but this lab in particular that got in trouble for this kind of stuff or created was a player in the initial gain of function research stir, was going to be very careful about this, and they, I guess to their credit, they followed all the rules. They
could have removed the change, right, and see if you abolish.
Exactly, you could do that. You could do the reverse experiment.
I would say it probably wouldn't fully. There are probably other changes that we don't know about.
Well, and I think that those points are exactly some points that I was thinking about at this point of the paper, because I was thinking that, how much time has the virology community collectively spent on thinking about influenza and influenza pathogenesis? It's relatively well studied out of everything. And we have the sequence of this virus, and yet we can't point to residues that are involved in these pathogenicity changes. We can't predict exactly which parts. We can say, yes, this one particular amino acid change in polymerase seems to be potentially important, but we don't know, as Vincent points out, if there are other changes that are also important in other genes. And this is a really, really well-studied virus. And so to me, the ideas of, that we would know enough to make sort of changes to do some of the nefarious things that people are worried about. Really? We can't even figure out which base pair changes are important in this virus that we've been studying the most of all the viruses.
There you go.
The other thing I was thinking, following up on my wrap, is that whether or not you could actually do the animal experiment with the gain of function virus. This particular mutation is nevertheless definitely on the radar. Okay, so if the whole idea here is that we try to look at characteristics of viruses that have pandemic potential, and this is a suspect characteristic, this particular mutation.
All right, so the last thing in the discussion, they say, okay, this Texas virus is lethal in mice and ferrets, yet the person who had it was fine. So why is that? And they say there are a couple of reasons. One is that maybe we're really looking hard for, and we find mild cases that we didn't find in the past. Okay, but if there were severe cases, we would see those too. We don't see any, right? So I'm not sure how important that is. And then they say eye infection with a low dose of virus may just give you less severe disease in humans, and there's no guarantee it's going to go from the eye into the upper and lower tract, right? Where you would need it to go to cause severe disease. And then they say maybe people have some protection against H5 from the multiple infections with seasonal influenza viruses. We don't know this. So, but that's the main caveat of this paper. I don't understand why in animals this is so bad, and yet in people it seems to be okay.
Ferrets aren't humans. You said it yourself.
Yeah, they aren't. They aren't. People are always, I'm so worried about H5N1. And I think, we know it's there. We know it's circulating. It's probably not going to be a problem because the viruses that are a problem are the ones that, boom, all of a sudden they're there and that's it, right? Like 2009 H1N1, the last flu pandemic. Few cases initially, and then in a week later, boom. And we'd never seen that virus before. So
I'm worried mostly about driving to the store.
Someone wrote the other day, and this is a good point. Before they abolish infectious disease research, we better make sure we have rapid antigen tests for H5N1 viruses, right?
So I was pasting in here to the show notes. This two things. One is a worldwide compilation of cases of H5N1 reported in humans. And then the other one is this table. I don't know if that link is working for you guys, from the WHO, which breaks down by country over time from 2003 to 2004, number of cases and number of deaths. I'm still highly intrigued by the fact that they saw H5N1 PFU in all of the organs, all of them, just like everything they checked. So has anyone checked in humans? Does this, because obviously people have died. I see it in the case numbers. Back in between 2015 to 2019 in Egypt, 43 people died from H5N1 and so on. So
they have looked, people died. They can look in various, they have multi-organ involvement, which is very different from seasonal influenza, and the virus is reproducing in these other tissues. Yeah.
Wow.
So it's a highly unusual influenza virus in that sense. Okay. So that's really cool infectious disease research, folks. But think twice if you want to eliminate this kind of stuff. Now, because Jolene is here, we're going to have now a phage paper, right?
But Brianne's here too, so it's going to be innate immunity.
I was so excited to see this paper.
This is, I had a really hard time picking so many great papers linking innate immunity as being studied in animals or in mammals, let's say, with innate immunity in bacteria. It turns out that there are many similarities, and that's where this paper comes from. So this is a paper from Cell. It was published in October. There are 4 authors, Samuel Hobbs, Jason Nomberg, both of whom are postdocs in the labs of Jennifer Doudna and Philip Kranzich. And the paper is titled Animal and Bacterial Viruses Share Conserved Mechanisms of Immune Evasion. So in this, and they are located at Harvard and at Berkeley. And in this paper, kind of the big picture is that we have seen similarities between the defenses that bacterial cells use and mammalian cells use against the viruses that infect them. Things that we didn't, there's just been a huge explosion of discovering systems that bacteria use to defend against the phage infections that they encounter. And this is where things like CRISPR came from, things like restriction enzymes. These were technologies that revolutionized our ability to create medicines and to do studies on infectious disease, but also chronic diseases. If we didn't have these technologies, we would not be able to do most of the biotechnology that is currently available today. And we wouldn't be able to make the leaps and bounds that we're making now that we couldn't make before. So turns out that in bacteria and in mammalian cells, the viruses that infect them also have ways of getting around those defenses. And we know that they have those ways, but there's a lot of diversity in them. So we hadn't seen a lot of similarity in the way that animal viruses get around host defenses and the way that bacterial viruses get around host defenses. So in this paper, they're looking for conservation in the way that the viruses get around host defenses between these very different cell types, bacteria and mammalian cells. So Or let's go animal cells. I have to say animal cells.
Yeah. Yeah, because they do a little bit of work with the Drosophila
Right.
receptors here too.
Yes. Okay. So I'd like to just kind of give the punchline here is that they found that there are these kinds of pox viruses that infect animals and phage that infect bacteria that encode the same kind of enzyme, a protein that helps them evade innate immune defenses. And those enzymes are so similar to each other structurally and enzymatically that they can actually replace each other. They can put the enzyme from the animal virus into the bacteriophage genome, and it works in a bacterial cell. So that's the main takeaway here. And now, the surge of interest between eukaryotic and prokaryotic innate immune systems has really revealed a lot of interesting things in terms of adaptive immunity, and that's what I was mentioning in terms of CRISPR. It's also revealed interesting things in terms of innate systems, which I would term restriction enzymes, which are proteins that recognize specific DNA sequences and cleave them. And there are a lot of similarities, but there aren't good examples of those with cross-kingdom similarities. So bacteria and animal cells are completely different. For the those who haven't taken biology, let's say, out there in a long time, the animal cell is huge and compartmentalized. The basic processes of DNA and RNA and making proteins, that's all there. But where it happens and how it's regulated is different from bacterial cells, which are much smaller. They don't have as many internal compartments, although they can subcellularly localize their processes. Now, one thing that is present in all of these cells is nucleotides. So nucleotides, remember, are the building blocks of nucleic acids like DNA and RNA. And they're there all the time. And they're not just used, however, for building nucleic acid. We know ATP and sometimes GTP can use for energy, but they can also, since they're available and around all the time, be transformed from the form of the mononucleotide that would be used for incorporation into nucleic acid into forms that are linked together, cyclic forms, where you can either cyclize a single nucleotide and then you can cyclize 2 or 3. So you can have dinucleotides or trinucleotides. And when this happens, these become signaling molecules because the cell only forms those when they encounter a signal that they should respond to. And usually those signals are things like infections, although it turns out that in animal cells, those can also signal danger from something like cancer. So abnormalities in the cell trigger these responses in the cell to cyclize nucleotides, and then they can turn on other defense systems that might cause the cell to die, which we call, in the case of a phage, an abortive infection. So that will kill the individual cell, but it may then prevent other neighboring cells from being infected. Sorry, I
was going to say one other thing about cyclic nucleotides.
Yeah.
Because I think that, when I got very into this particular pathway that we're going to be talking about, I started thinking a lot about cyclic nucleotides. And one of the things I didn't totally realize was that I actually already knew about cyclic nucleotides in a couple of other situations biologically. And I didn't really think about the ways that they were all connected or the ways that there might be sort of similarities. And as you said, that this is sort of a general theme that cells are using for signaling. So if listeners have thought about sort of general cell biology, they might have heard about CAMP. Cyclic AMP, that's generally used in signaling. That's a cyclic mononucleotide. And they also might have thought about quorum sensing, particularly in, say, Pseudomonas, which often uses a cyclic dinucleotide for as its autoinducer. And so I found it really interesting when I started getting into CGAS, when I started sort of getting into the field, to think about the fact that at first, this seemed like a really weird idea these sort of modifications of nucleotides, but in fact, we had already known about them and maybe hadn't put it together as like a big class before. But you may have heard of them in other places in biology and just not realized that was what was going on.
Yeah, good point, Bran. So the particular pathways that we're looking at in this paper are the cyclic GMP-AMP synthase, or CGAS, stimulator of interferon genes. So that stands for STING. So CGAS STING. And then that's the name for the pathway that's found in humans. And there are similar pathways that are found in other cells. So they're called CGAS-like receptors, or CGLRs. And then it turns out that in bacteria, there are CGAS-like enzymes. They're called CGAS/DNCV-like nucleotidal transferases, condensed down to CDNTases. And I'm not going to use a lot of these abbreviations throughout the paper, but there are classes of them because there have been many identified. And so these are known to cyclize GAMP or GTP-ATP into either linked in different ways. And those then can activate the, in bacteria, system called cyclic oligonucleotide-based antiphage signaling systems, or CBAS. CBAS. So
I've never been able to be on board with that, but that's okay.
So these systems, they sense infection and then they can turn on cell death pathways. And the production of the signaling molecule, the cyclic nucleotides, will amplify the signal that they have detected an infection. So the enzymes that do this, that's what we're interested in this paper. And at the start of this, they knew about a particular protein called ACB1 from phage T4. And T4 is a large double-stranded DNA phage with a lot of genes whose functions we do not know. So recently, just a couple of years ago, the first author in this paper reported the ACB1 protein and how it could cleave this dinucleotide signaling molecule.
Jolene, I have a burning question.
Okay.
Sorry to interrupt.
Yeah.
But I'm trying to review for myself. In mammalian cells, the CGAS, the enzyme that makes the cyclic dinucleotide, if I recall correctly, is induced by DNA in the cytoplasm.
Yes.
Right?
Yes. So
DNA in a eukaryotic cell shouldn't be in the cytoplasm. So if it's in cytoplasm, something's wrong. Mm-hmm. So you induce this enzyme, and I forget the details of that, but I don't really care as long as that's okay.
Yeah.
What's the story in bacteria? Because in bacteria, you don't have a nucleus, okay? So somehow the bacteria must be able to discriminate between an invading DNA and their own DNA.
Yeah. Yeah. We don't actually know all of the signals that lead to this being activated in bacteria.
Okay.
And actually, that makes me bring up, I forgot to mention it before, there's a great review that was published earlier this year by Hannah Levidna and Aaron Whiteley on an overview of conservation and similarity of bacterial and eukaryotic innate immunity in nature. And in there, they go through all the principles and kind of lay out the similarities between these pathways. And if you look at the figure there, you see there's a phage kind of infecting the cell, and then there's a dashed line with question marks because we don't actually know what they're detecting to activate the CBAS system.
Stuff to do. This is great.
I'll tell you, Rich, the first time I read one of the CBAS papers, that was my burning question the entire way through the paper.
Okay.
And I found the author's explanation to be wholly unsatisfying. And I have sort of had that question and wondered that about this every time I read a paper.
All you got to do is say, I don't know. Or put a dash line with a bunch of question marks. Cool.
Yeah.
Well, while we're paused, I wanted to point out that they're going to talk about this phage protein ACB1, and for various figures, they start talking about T4. And so it's very late in the paper when they finally connect T4 and ACB1 in the same sentence.
Yeah.
So by then, I had already looked it up in another paper to figure out what phage it was because I'd seen this figure that had T4 in it. So that was a thing that was a little missing. But if somebody takes a look now, they'll know.
Yeah.
Now, Rich, one last thing. Not only did you work on poxvirus, but didn't you work on T4 as well?
T7, and I was going to give this rap if we got to it because this paper pushed all of my buttons, okay? Because I cut my teeth on T7, and your first love, you just don't get over it, all right? And I could have done that. I could have done that forever, but felt the pressure to become eukaryotic. And a little known secret is that poxviruses are just big phage. Okay, so that's what got me into poxviruses.
Nice.
And this proves it.
So the connection for the authors between seeing this ACB1 enzyme, that's a phosphodiesterase that cleaves that cyclic dinucleotide, and the pox viruses that we keep bringing up, was that they had solved the structure of it and seen some important aspects of its activity. And there was another paper by the second listed author on this paper which came out earlier this year. They published a database of eukaryotic proteins and their structure predictions, 4,000 eukaryotic viral species and their proteins. And I just have to say, I'm really annoyed a lot of times. Everybody's like, oh, AlphaFold, it's so great. AlphaFold 1, 2, 3. But they don't predict viral proteins in all of their databases and so forth. It wasn't trained with viral proteins. They're not available. So Viral Zone recently posted the predictions for most of the viruses that infect humans, and they have the predictions for those available, which is great. But that doesn't include things like what are both of the subjects of this paper. So what they did was they took that structure and they found homologs of ACB1 in this database. So structural homologs, not necessarily related by sequence.
This blows my mind. AlphaFold blows my mind to start with, okay? You can predict structure, and then that you can compare structures. They may not have any, very little, or at least not recognizable sequence homology, but you recognize structural homology. I don't know how you do that. I believe it, but I don't know how you do it.
So I've seen that come out of the Convergent lab earlier in terms of the structural prediction. And it's amazing computational work that I am so impressed by, but that's actually how they found this entire family of CGAS-like receptors in Drosophila, is that they did structure predictions and found structural homologs that they wouldn't have previously expected. So it's a whole thing that the Convergent lab has been doing really well for the past couple of years.
It just occurs to me that, this protein structure stuff, prediction and comparison, may be as big a paradigm shift as some of the nucleic acid stuff that we've seen happen in the past, like cloning and sequencing and the rest of it. It's huge.
Well, it got a Nobel Prize.
Exactly. It just got the Nobel Prize this year. Okay. They found homologs to ACB1 in that database, and one of the proteins that they found is in a pox virus, in particular a penguin pox virus. So they took that particular protein and they solved the structure of it, and in various forms. So in the first figure, they're showing us the structure of the pox virus with the pox virus protein with its substrate, and comparing that to the phage protein, ACB1 protein, with its substrate. And I think it's really beautiful. It's like a cup of beta strands, and in that cup is where the substrate is. And outside the cup, there are these, they call them stabilizing alpha helices that are keeping that together. And then each of them has what they call a lid domain, which we will come back to more later, but there's something that kind of clamps that cyclic dinucleotide in the pocket, in the cup.
So, Jolene, they already had the structure of the phage protein, is that right?
They did, yeah. Okay. That was a couple of years ago. They published the paper about ACB1, which included the structure. Yeah. And so here, they show us the structure. They see all these similarities that they had detected using the structural similarity comparison. I'll just comment on that. So my understanding of how it works, it's a lot of math. You have the predicted structure of one, the predicted structure of the other, you superimpose them, and then you calculate how close everything is to it and assign it scores. So that's how they then, I'll just skip ahead here. So they took the structures of all of these phosphodiesterases that they could find for viruses that infect prokaryotes and eukaryotes, and they plotted their similarity scores, and they see that the ones that are in the prokaryotes separate pretty well from most of the ones that infect eukaryotes, except for the rotavirus proteins, and rotavirus proteins cluster more with the prokaryotic viral sequences. So that was their first clue that these were probably not coming from the same place. Although they have structural similarity and we'll see functional similarity, they did not likely come from the same original gene.
So like a convergent evolution.
Convergent evolution.
Mm-hmm. So the structures that they solved show us the active site, which includes the amino acid residues that bind to the substrate and are involved in the chemical reaction to cleave the substrate. And they see that they're right on top of each other, and they're pretty much the exact same residues as well. So they then used these really kind of neat tools. So you take an analog of the substrate that is resistant to cleavage by the enzyme, and then they solve the structure of the protein with this analog in the binding pocket, and they're able to get differences that would occur before binding and after binding and in the middle of the chemical reaction that cleaves the dinucleotide. And they see that there's a lot of similarity in how the penguin pox protein and the phage protein are binding to the substrate. And then what they do is they make changes. They change the residues that are encoded in this catalytic site of the penguin pox protein. And they test each of those individual residues for activity, and they see that all of them are necessary to be able to completely cleave the product to its normal final cleaved product. And then they show us their hypothesized mechanism of the actual chemistry of the cleavage, and they check with a couple more mutations to see if that is consistent with their model for how cleavage is working. So here they have to bring in actually the lid domain. So the lid domain is on the way it's oriented in the paper anyway. They show it on top, and it is clamping down, as I said, on that nucleotide. So they've got the structure of the apo enzyme, which is without the substrate, and then the structure with the substrate. And I just thought this was really cool. There's a huge shift in the position of the lid domain. So in the pox protein, it is an alpha helix, and when the substrate isn't there, it's kind of hanging out in the field, and it leaves a big open spot. And then once the dinucleotide is bound, it shifts, they say, 25 degrees over to clamp it down. And the lid domain in the phage protein is actually different. So the lid domain in the pox protein is encoded on the N terminus at the beginning of the protein, but in the phage protein, it's encoded on the C terminus completely at the other end, and it's not an alpha helical domain, it's a loop that kind of stretches out over the dinucleotide. And they do delete that lid domain and show that, at least for the pox protein, when they remove the lid domain, they definitely are not having the correct cleavage activity. With the phage protein, when they delete the lid domain, they actually kind of get some variable amounts of activity. They get some cleavage, intermediates, and product as well as the original product not being cleaved. So the absolute necessity of that lid domain varies from one enzyme to another, but that is helpful in reaching the final cleavage product for both of them. Now, with the pox protein, where it's more important, they see that there are some residues that might be involved in the cleavage based on their position and conservation. So those are also ones that they then mutate. And the residues that are changed are shown to be important. If they're on one side where it binds to the guanine, or it looks like it would bind to the guanine or have some stacking interactions with the guanine part of the dinucleotide, but the ones that are on the side of the protein where it would be binding to the adenine are not as important for cleavage.
I love that they're using thin layer chromatography.
Mm-hmm.
Yeah. Yeah, I definitely don't see it very often.
But if you want to look at these small molecules and products, you have to use it, right?
Yeah.
And they're using radioactivity for it.
Gosh,
So,
yeah, they are.
That's right. I think that to be able to distinguish between the products, this is probably the quickest way, unless you're
Yeah.
going to be doing some sort of mass spec identification of them, or maybe HPLC.
Yeah, I think a lot of the experiments that involve some of the cyclic dinucleotides end up being techniques we don't see very often, because you end up needing to use that. And so it's always sort of cool to read some of those papers and then have to think about, wait, what's going on in this technique again? And when did I last hear about it?
You used to see them often, but you got to be old.
Yep.
They're good techniques for the experiments that they're showing here. And I think that's because TLC allows them the resolution to detect the differences between the intermediates. So you can see the cleavage products very easily. They show here what the expected conformation of a cyclic dinucleotide in the sting molecule is. So the host enzyme that detects the signaling molecule and then initiates the defense pathways. It's this kind of nice like U shape. But once that cyclic dinucleotide is cleaved, for instance, by the phage or other virus protein, then it loses that shape completely. It's linear. And so the sting molecule or other sting-like molecule probably wouldn't recognize it anymore. Now, then the last set of things that they do, I think this was great. They went back to the databases of viral proteins and looked for more structural homologs. And here they brought in a Drosophila virus protein. So this is Drosophila. See if I can remember the name of the Drosophila
It's an
virus.
iridescent virus of some sort. I forget.
Yes,
exactly what it was.
invertebrate iridescent virus 6. And that also has a protein with some structural similarities. It has the cup predicted and those bracing alpha helices predicted. And so they took these 3 proteins, the penguin pox virus phosphodiesterase, the invertebrate iridescent virus phosphodiesterase, and the T4ACB1 phosphodiesterase. They purified all of those, and then they took a panel. Now, I'll read out just a few of the substrates. They have the 2,3-CGG or 2,3-CGAMP, and then they have a series of 3,3-CGAMP and other variations. And so exactly the names I don't find as important, that there are a series of them that are found normally in animal innate immunity signaling, a series of them found in bacterial antiphage defense signaling, and then a couple that are just normal host signaling molecules. And so you wouldn't expect the phage or the virus or the ones that are defending against a defense to cleave those. Only they should match. They should have some matching. So what they did was they took that whole panel of the potential substrates and the purified protein, and they did a dose response of cleavage assay for each of them. And they saw that the penguin pox enzyme preferred the substrates that are used in animal antiviral signaling. The phage ACB1 preferred substrates that are used in bacterial antiphage signaling. And the Drosophila virus, I don't know if I'm going to say like that it has a preference actually. It kind of cleaved quite a few of them, but if there was one that was more similar, it was more similar to the animal antiviral signaling ones.
Yeah. So I think that I thought that this was, particularly interesting in thinking about some of these. So if aficionados are wondering, the numbers and things are those same numbers that we use when we talk about DNA structure, and the 5' end and the 3' end based on those carbons. And so this is basically, are you doing like this or like this or like this?
Right.
With which carbons are getting joined together in the cycle? Is it the 2' and 3', or is it the 3' and 3' on the two different molecules? But I think that, I had first learned about all of this with C-gas and mammalian cells, and C-gas is predominantly known for making 2', 3' CGAMP. So that's a G and an A. As opposed to some of the others, which are GG or AA. So it's a G and an A bound with a particular linkage. Mm-hmm. And it was the first time, I started reading more about this, it was surprising that there were even others that were made in bacteria or that were made in Drosophila. And I think that this experiment made me really think about this more as an arms race, that perhaps one of the reasons why C-gas is making this unique product, 2', 3' CGAMP, is perhaps because of evasion, that maybe viruses were able to evade some other products. And so that maybe, all of these different variations of cyclic dinucleotides were needed because earlier versions of these enzymes could cleave different dinucleotides. And so the biochemical linkage needed to change to try to evade some of these enzymes. And I hadn't really thought about that and why 2', 3' CGAMP was unique until I saw this figure and read this part of that paper.
Yeah, I think that's another part of the argument for the importance of the lid domain, because it really allows that enzyme to clamp down on, and actually I didn't talk about it, but it allows it to torque the cyclic dinucleotide into just the right position for those catalytic residues to catalyze its cleavage.
Absolutely.
Mm-hmm. And so then the last experiment, which was the thing I was very excited about seeing in this paper, was they said, okay, we've got these proteins that in vitro biochemically all have the activity that we would expect might confer activity in vivo, but in a cell, when these actually get turned on, we want to know, will these replace each other? And so they did engineering of the phage T4 genome to both, first of all, remove a second copy, you could say, of the gene. There's another ACB gene in that genome, and it doesn't cleave cyclic dinucleotides, but it binds to them and kind of acts like a sponge so that they can't be detected by the host. So they took out that gene and they left in only the ACB1 gene. And then they had a version where they inactivated the ACB1 gene. And then they replaced the ACB1 gene with the penguin pox virus phosphodiesterase gene. And they first showed that that it replicated normally. They also showed that it had activity in a cell lysate, that the penguin pox virus enzyme could function in a bacterial cell, which it does. And then they show that when they turn on a CBAS system, that it will usually inactivate the T4 infection. So when there's no ACB1 gene in the genome, you have a much lower number, about 100-fold to maybe between 100 and 1,000-fold less plaques being formed. And with the active ACB1, you have 100 to 1,000-fold more plaques being formed. And then with the penguin pox virus phosphodiesterase, you see essentially the same thing as you see when the phage ACB1 protein is present. So that suggests that the animal virus protein that normally is defending against these cyclic dinucleotides is able to function from a phage genome in the bacterial cell.
That's very cool.
Mm-hmm.
This is showing that there is much more similarity in the much more convergence on the ability to evade host defenses than we previously could recognize, because there's not sequence similarity. And I think this wasn't a given, because the diversity of defenses against antiviral pathways is enormous. The anti-CRISPR proteins all do vastly different things. So there's no guaranteeing that these were going to be similar to each other. However, here we definitely see that they are similar. Now, what they didn't do, and I guess I after thinking about it, I could see probably why they did not put the phage gene into an animal virus and see if they could get the reciprocal experiment to work.
Right.
I'm like, okay, well, maybe you don't actually have the penguin pox virus and a system to grow it in.
Right.
But I think it would be great to see these phage proteins functioning in animal cells as well.
Yeah. And I think that would be really interesting in terms of because animal cells are using the 2', 3' CGAMP more predominantly, would it actually work that way? Or has the, sting sort of specialized enough that it can't work on, that it can't do that cross in the same way?
Yeah.
Would doing it in Drosophila be an easier way to get at it than maybe doing it in the pox, or do we not know enough about the Drosophila system?
So the Drosophila system, again, so that one is more like 3', 2' CGAMP. So that one again has its own CDN. I don't know enough about the Drosophila system, and particularly don't know enough about this virus that they're using in Drosophila to know.
So I've been playing on the side while you guys are yakking here, because I was thinking to myself, anybody who is going to snicker at the notion of studying a poxvirus of penguins should pay attention. And then I decided, well, now wait a minute. I don't know anything about penguin pox. So I looked it up, and I think from what I can see here, that it wasn't really identified and certainly wasn't sequenced until 2021. And it's not like somebody going out and just isolating viruses and sequencing them for no particular reason other than to have the sequence, which is a good thing to do, okay? But it turns out that there is a very rare penguin, what's he called? Yellow-eyed penguin in New Zealand that is has a major problem, which is a disease called diphtheritic stomatitis that it turns out is caused by this penguin pox virus.
Oh, no.
That's a threat to the species. Pox viruses typically cause these nodular or blistering lesions on the skin. This happens in the trachea and causes a diphtheria-like disease in these penguins. So these investigators went out, isolated this virus and sequenced it, and that's where penguin pox comes from.
Huh.
Whoa.
That's cool. See, I read this paper and I think, wow, the fact that there has been this convergent evolution to deal with cyclic dinucleotide mediated immune responses means that those cyclic dinucleotide mediated immune responses must be pretty important and strong and a pretty big problem for viruses. So that viruses across kingdoms have had to evolve around it. So I'm excited that I spend time thinking about them because they must be important.
Furthermore, what blows my mind is that a convergent evolution. So we're talking about, okay, you got two groups of organisms faced with a similar problem that has in common this dinucleotide or trinucleotide structure. How are you going to deal with this? And apparently, so far, independently, they come up with a structural solution that is remarkably similar. Okay? That actually makes me, I have to say, suspicious that it's really convergent. And I wonder whether back there in the soup, okay, there isn't some commonality or something. If not, that's a really remarkable convergence.
They do acknowledge this caveat in the limitations section that I think is a requirement of the Cell Papers. So they say that, they can't actually determine any kind of evolutionary relationship or lack thereof. And I think it's based on, they just don't have a lot of data. They didn't have this database released until earlier this year. So having more examples to compare to each other will allow us to have a more clear understanding of the relationships between these kinds of enzymes.
There's not enough dark matter in this particular realm. Get out there and sequence. I don't care what it is.
So I just want to give like a brief recap of the paper. So there was this, the authors had this question about whether there were functional and mechanistic similarities between how animal viruses and bacterial viruses get around the defenses that their hosts put up in the cell. And they found that there are enzymes, proteins that are so structurally similar, they have very likely very, very similar chemical mechanisms of cleaving the signaling molecules that the host makes in the cell. And they're so similar to each other that they can replace each other in the other kind of cell. And why this is one reason this could be important and useful is because bacteria in particular are very amenable to fast and cheap studies. And so we know that these are really important in our cells, not just for defending against viral infections, but for normal cell processes like when you have the potential development of cancer. These are the kinds of things that cells use to signal to your body that something is wrong. So having other systems to use to both define the intricacies of the mechanism and make comparisons that let you make new insights into how they're working can really potentially give us new ways of developing medicines for human diseases.
Awesome, Jolene. Great choice. This is good stuff.
It's infectious disease research. That's it. It's done. All right, let's do some picks of the week. Brianne, what do you have for us?
On the previous episode, I had mentioned I've been getting really into a space podcast, so I figured I should say what that space podcast was. And so I've been listening to something called Pale Blue Pod, where a woman who has a PhD in astrophysics as well as a degree in folklore and who also does some kind of science fiction writing, talks about some sort of physics or space related topic once a week. And I have learned a tremendous amount and feel like, if nothing else, I could ask much more educated questions than I could before. And really, there are topics in here that I feel like I have read or thought about for many years. And in her explanation, I've suddenly been like, oh, that's what that is? I get that now. Like, how did I not get that before? So I've been really appreciating it. It's been a lot of fun. And in fact, it also makes me think a lot about things we do here on TWiV and sort of, boy, I hope that our listeners are getting that same experience.
How long are they?
About an hour.
Okay. Man, does she do them all by herself?
The first.
Oh, she's got some guests.
The first 100, she had a co-host, and now the co-host has moved on, and so she has a guest each week.
There's somebody here called Tribble.
That was some comedian who I had not heard of before the episode.
All right, I'll have to.
There is a quote on one of these pages. Always remember that you are space.
That is actually their tagline as they leave. The beginning is that it's the podcast for people who are intimidated by space but want to be its friend. And the end is always remember that you are space.
Space. The kind that make you sick. Kathy, what do you have for us?
Well, first of all, speaking of physics, in the past we have picked videos by Physics Girl, and unfortunately she's had a very severe case of long COVID. And so she recently posted something on Instagram that was marginally hopeful. She's able to post something and say a few things that she's doing, but she's still bedridden and so forth. So
wow.
It's just really makes you think about why, those people who aren't worried or scared about the virus, and so they're not going to get vaccines or they're not going to get antivirals, should maybe think again. Anyway, but for me, it was encouraging to see that. And then I've mentioned a couple times various things that I found over the last couple episodes, and they all come from this thing called Science Advisor, and you can sign up for it. And so that's the primary pick for today. It's a free daily science news distillation by science, and they give you things from science and they give you things from other journals. And so it's really broad. And so just, between the last time I was on and this one, I already had 4 different things that I wanted to pick. And so I'm just going to mention them to you, and then you can go find those through Science Advisor, or maybe if Vincent puts the link in, you can go back to the original article. But one is about semiconducting hydrogels. So hydrogels are important in biomaterials science for things in human health. And so this is a way that they've now been able to combine the best aspects of self-conducting and hydrogels. So they're soft, flexible, and stretchy, and they can conform to wrinkles on your knuckle, for example. Another one of these stories was about a bot that can pick up things because it has a soft touch, and they were inspired to figure out how to do this picking up items by something called a velvet worm. Another one was about a bot that can fly, 2 bots that can fly stably. One is called PigeonBot 2, and it has a mechanical skeleton with real pigeon feathers. And then the second one is called LizRaptor, and it's a drone that's been inspired by a hawk. And it also has adjustable wings and a movable tail.
Wow.
And then the last one I just thought was a really nice piece of good news about a way to zap forever chemicals like PFAS with light. And there were 2 different papers that they referred to about that. So it's just, to me, nice, interesting, optimistic things about what we learn from science, very current science. And they always have the link to the primary article, and they usually have a nice sort of lay summary in the newsletter itself. So you can pretty much get what you need just by reading the newsletter. So the Science Advisor sign up is free and I think really worth it.
I guess PFAS is what Martin Macari's talking about, chemicals in our food supply.
Well, yeah, I mean, the PFAS are forever chemicals, and they're the kinds of things that, made things stain proof, like Scotchgard back in the day, or, that protected your carpet or your upholstery or things like that. And So.
But are they still used?
I think in some cases they are.
Yeah, I think so. Yes.
All right, well, Marty will get rid of them. That's good.
Yeah.
And we'll have stains everywhere. No problem.
Yeah.
Rich, what do you have for us?
So I've picked this before, and I can justify the repick. It is that time of the decade. Every 4 years, the Vendée Globe happens, and this version of the Vendée Globe was launched 19 days ago. The Vendée Globe is a sailboat race where the challenge is that it is single-handed, so there's only one person on the boat. It is nonstop. If you stop, you're disqualified. And it's around the world. They start in France and sail south. The leaders are now, after 19 days, just rounding the Cape of Good Hope, and then sail through the Southern Ocean past New Zealand and Australia, and back up the Atlantic back to France. It's about, I don't know, that's like 25, 30,000 miles, something like that. Takes them, the leaders finish maybe after 3 months. There are 40 individuals in this particular race, one who has already had to drop out because of mechanical problems and an injury. Actually, that's pretty remarkable that there's only one dropped out so far. So the leaders are just entering the Southern Ocean, which is where most of the real excitement happens, because the Southern Ocean is not a hospitable place to be. These are amazing boats, 60 feet long. They're designed 60 feet.
Wow.
Okay. So they're designed like, it's almost like they're huge surfboards. And now, of course, they have wings, these, modified dagger boards that give them lift and add to the speed, and all sorts of other technologies. And so they're designed so that, in particular, when you get in the Southern Ocean, you don't fight with the waves, man. You use them. You're just surfing down the surface of these like 40-foot waves. It's amazing to think about what they do. And the website over the years has become more and more sophisticated. So I linked the website here so they got a tracker where you can see where everybody is. You can get animation of how they got there. You can get information on the individuals. You can get information on their boats. They have interviews with these people. They do satellite interviews, so you can see what it's like to be on the boat and all that kind of stuff. And a lot of them actually are collecting data as they do this, okay? Because the Southern Ocean, not many people spend much time there. And so there's a lot of people that are collaborating to collect data on the Southern Ocean while they're doing it. So get hooked. It's good fun.
How many people are on these boats?
Just one, single-handed. Single-handed, round the world, nonstop.
So do you sleep?
Yeah, you learn to basically sleep by catnap. Have you ever read a book called, I ought to pick this, maybe I did sometime back there, Never Cry Wolf by Farley Moat?
No.
Oh, man. Okay, I'll write this down as a pick. There was also a movie made. It reminds me of it because of what he did. I'll pick it another time. He learned to, basically catch his sleep like a wolf, okay, which is not you get 8 hours. It's like you get 15 minutes every hour or something like that. So these guys go down and they catch 15 minutes, and the boat's got all sorts of alarms and stuff. So if it goes off course or some weather event is going to happen or something, it wakes them up.
So did they go below to sleep?
Yeah.
Right,
they got a bunch of autopilot stuff.
Oh, they got autopilots? Okay.
Sure. Yeah. These are really high-tech boats. So I'll tell you, they got similar races where the one of the mandates is you got to use 50s technology. Okay. So in terms of autopilots and stuff like that, you're talking about wind vanes that keep the boat oriented to the wind properly. That's even scarier than this stuff, okay? But there you go, yeah. Sleep,
so
good question.
These are sponsored by something, right?
Oh, yeah, this is big money. Okay, so these boats all have sponsors. These are professional sailers.
So many of them are from France.
Yes,
it's
because
the vast majority.
Yeah, the race originated in France, so a lot of them are French. There's only a couple of Americans. There's as time has gone by, there's increasing numbers of women. There are several women in this race.
It's all sail driven, right? No motors.
Absolutely, yeah. If you start your motor, you're disqualified.
And what if the winds go away?
then you bob around for a while.
Because you
that-
said if you
and
stop, you get disqualified.
you can't anchor. Okay, you can't. I think no, you can't. You can't anchor or contact other humans or have help unassisted. That was the other thing. You can't have any assistance other than radio communication or satellite communication with your team. Okay, that can help you with stuff like weather and that kind of stuff. The website. If you look at the tracker, there's a bunch of different overlays. So in particular, I like seeing the wind. If you click the wind overlay, you can see what they're up against. Okay.
You would have to download a lot of podcasts and audiobooks.
Yeah.
And they have enough food for this journey?
Yeah.
Oh my
And they're
God.
not doing any fishing, trust me. Okay.
Probably not cooking anything. Probably everything is in a package, right?
Yeah, well, it's packaged stuff, but you can. I think they must have desalinators on board so that they can make water.
Mm-hmm.
Okay.
Hmm.
And so yeah, dried stuff, but you hydrate it and cook it up and that kind of stuff. And they have some, you see them do celebrations. Like I'm sure that there's some Christmas videos in here. But yeah, so it's fun to follow, and it's a good long effort.
I remember you've picked this before.
Yeah,
and
right. I
still
pick it
think
every
it's
4 years.
Just so crazy. So crazy. But, humans do amazing things, right?
Humans do amazing things. And I'm glad you said that because it can be depressing to see, what's going on in the world. People are still killing each other. There are serious problems of climate change and stuff that humans need to do something about. And it's depressing that we get ourselves into this mess. But on the flip side, we do amazing stuff. Just incredible stuff. And we have the capability to get ourselves out of these jams. And we probably will. It'll probably take a crisis, all right, unfortunately. But, it's good to celebrate the good stuff we do. Yeah.
Some people say that only the capitalist societies make the really good stuff.
Look at the
those people are
transportation
wrong.
system. Those people are wrong. The Chinese are doing some pretty amazing stuff. Okay,
I don't know. I have no idea.
The high speed trains. They've got. I saw an article the other day about a new public transportation vehicle that is basically a trackless streetcar that follows just painted markings in the road instead of having tracks.
Wow. Cool.
Okay. I won't ask any more questions. Charlene, what do you have for us?
I have something called the SEMER Project. This is the Center for the Improvement of Mentored Experiences. It is a project that has collected a series of evidence-based practices for training mentors and mentees on the best practices for those kinds of relationships. And a lot of the work that is in this program is funded by the National Institutes of Health to help both the people who are training the up-and-coming generation of scientists, as well as scientists in training who find themselves in scenarios where they need to help their mentors mentor them. So I have done some of the SEMER project training through the on-campus offerings that we have here. This is the kind of thing where you can become a trainer that has been trained in the SEMER curriculum, and you can then go back to your campus and offer these courses, or you can have the SEMER team come to your campus and talk to typically PIs, go through the mentoring training. So professors and other scientists who supervise trainees. It's primarily based on STEM paradigms of careers. And then there is a set of curricula that are for mentees that did not exist when I went through it. But these are all about things like, how do you have difficult conversations? How do you talk about the thing that you want to do in the future? How do you help your mentees set smart goals that are realistic and time-bound and help them actually get where they want to go? And how do you How do you help them network? How do you help all the kinds of skills that STEM trainees need to develop? How do you equip them with what is necessary for them to do it? And how do you proceed when things aren't going the way that you would like them to go? And then for mentees, it's a series of skills to help them with setting their own goals and finding more than one person that can help them meet those goals. Because as I love the saying that I heard from someone named Carrie Ann Rockamore, there is no mentor, there's no guru mentor, no guru who knows it all. But a lot of times in our scientific training processes, we kind of treat the person whose lab we trained in as the person who knows everything and who should give us all those skills and pieces of advice that we need to get where we want to go. But that's not really the most realistic or maybe the most efficient way to do things. So on this CEMER project website, they have access to scheduling these things during the year. They often will advertise where they are doing an in-person training. They also have some free resources listed that are kits, things like examples of mentor-mentee compacts and links to other organizations that can help you with the training. My favorite one there is the NRMN, National Research Mentoring Network. And so I highly recommend it. And the reason I've been thinking about this a lot is because I just think about how the future generation, when I was a trainee, I was really lucky to go to a good school that had access to a lot of professional development workshops. But not everyone is so fortunate. And being given the ability to expose yourself to the knowledge and the structures and the framework for thinking about how to get what you need to get where you want to go is really good, because our graduate training programs in particular, but definitely undergraduate training programs, do not equip students with these skills in an even manner. There are some programs which are great. There are some programs which are really great for a short period of time. And then there are some programs who just are on their way to getting to great, to helping the students with that. We put a lot of professional development requirements on the students themselves. We say like, well, if you want to do this, then you have to go out and find these things. So having a structured way for them to approach it, I think is a really good thing for us and for them. So that's why I pick CEMER.
Thank you so much. I'm so looking forward to going through this website.
Yeah, it sounds really
This
good.
is all good. And I have to say, I went to grad school in 1975. None of this existed.
Yeah, right.
None of it. What do you want to work on? Okay, see you tomorrow.
Yeah. You're on your own, dude.
It's like my friend went to another lab. He said, what do you want to work on? I want to do this. Okay, the columns are in that drawer. And he went back into his office. The columns are in that drawer.
I think it doesn't really change the fact that you still have to be, you have to work hard, and you have to be curious, and you have to advocate for yourself. But I think that it helps even the playing field for some people who aren't in situations like that, and for people who are in situations where a lot of things are handed to them. Kind of takes a little bit of the luck out of your training path. Yeah.
No, it's good stuff. All right. Okay, my pick is a news article in Nature by Miriam Nadaf. It's called "These Two Ancient Human Relatives Crossed Paths 1.5 million years ago." I kind of like human. I don't know what's the field. Anthropology.
Yeah, it's anthropology.
Because you make so much of so little, a couple of bone fragments. This one, there are these two species which are parallel evolutionary on the evolutionary tree of humans. They're parallel to us, and they're not. We have a common ancestor, but and that's Homo erectus, which is a Homo species, and then we have Paranthropus boisei, and they found footprints of the two of them pretty near each other. So these are 1.5 million years old, and the footprints are, what is it? They're 13 footprints, okay? They can tell how fast they walked by the spacing. of the footprints, and they were not too far apart, like 30 meters or something. So they have this lovely artist's impression of these two species like walking next to each other, like with all these animals around them, which probably never happen, right? Animals would be too scared or whatever. But and they also have footprints of animals that lived at the same time. 30 relatives of cattle, 3 horse-like animals, and 61 birds, including a giant extinct stork. So I think this is so cool. So this is the first evidence that these two species coexisted. They walked, and the calculation is they walked within hours or days of each other. So they weren't walking next to each other like the picture shows, but they were living at the same time, which you can, and you look at the footprint. Yeah, it looks like a big foot. But you do lots of measurements and you can get all kinds of cool information, like the range of motion, the angle of the toes, and all this stuff, and how they're different from modern humans. So I think this is very cool.
I would have liked to know another human-like species. It's too bad they're gone.
Well, Homo sapiens existed with other Homo species, right?
Yeah, right.
But like you
And we
said about
wiped them out.
humans, we probably wiped them out.
Yeah.
We're about to wipe ourselves out.
Yes. Yeah, well, the nasty ones will be left, Jolene. That's the problem.
Oh, God, what a thought, Vincent. Jeez.
It's a shame because of all this good stuff, right?
Yeah, right.
Well, the research we talk about is amazing. And these boats that the things that people do, oh my gosh, it's all so cool. And what a waste to destroy ourselves. But we have to be more
and the
positive.
problems are all the same. We went and saw the movie Wicked. I'd never seen the play.
Oh, it's so good.
So that the themes there that was written like 20, 30 years ago. Okay. But the themes are like it could have been written yesterday. And if you think about it, and I spend too much time thinking about this, it's not just 30 years, it's 2,500 years. It's as far as you can go back. The problems are the same. Okay.
I don't understand why we have problems.
Greed. Hatred
No,
and delusion.
Well,
I
a
was
lot of
just
it's
thinking-
religion, right?
Well, I was just thinking the answer to everything is money.
Money too.
It's greed, yeah.
And then why some people need to be dictators, right?
Greed.
And not let people decide
Greed, yeah.
what to do. I don't get it. It
Greed.
doesn't make any sense to me. But what do I know? I'm a scientist. Okay, we do podcasts about science, not about sociology and government and politics.
Are you
That's
sure?
probably good. Well, to an extent.
To an extent.
Okay, that's TWiV 1173. You can find the show notes at microbe.tv/TWiV. You can send questions, comments, picks of the week to twiv@microbe.tv. And if you enjoy these programs and you learn something from them, we'd love to have your support. You go to microbe.tv/contribute. Brianne Barker is at Drew University, BioProf Barker on Blue Sky. Thanks, Brianne.
Thanks. It was great to be here. I learned a lot. That's what I was supposed to say.
And you also learned that 2 hominids walked side by side.
I learned a lot of things.
You didn't know that before.
I did not.
You learned that people still do thin layer chromatography.
I did.
Do you have an apparatus in your lab?
In my lab, no, but I bet somewhere in the science building we do.
Okay. Kathy Spindler is Professor Emerita at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an Emeritus Professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time. This was a particularly good gathering. I really enjoyed this. Thank you.
Jolene Ramsey is at Texas A&M University, ramseylab.vercel.app. Thank you, Jolene.
Oh, thank you. And, just hold on. We're about to beat the University of Texas at Austin in a football game this coming weekend.
Ooh, is that right?
That's right.
Fortunately, I don't care.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.